Hello.
Hello. I'm gonna wait a couple minutes here, get started.
Are we learning about the eye today?
Mhm. Today is going to be solely focused on the anatomy of the eye, its functions, and then some eye problems, how the eye sees color, and how it's transmitted to your brain, and then we'll do some color theory, or go over color theory. Essentially, just Science Olympiad optics side day, basically. Okay, people still coming in. Yeah, it is recording. That's good. From for a disclaimer, I am not a bio person at all. I do purely physics and comp sci. Um, I do not do bio, so if you ask me to go deeper into the chemical, like biochemical processes, I am not able to answer those questions. I can tell you it happens. Going deep dive into why it happens is just beyond the scope of the textbook I learned it from, and thus I'm not sure I can explain everything. But there should be um, plenty to understand. We're gonna go deeper into anything that you deeper than anything you need for Science Olympiad if you're here for SciOly. If you're just here for the eye, still should be plenty of stuff. Just if you ask me, um, deeper chemical like molecules, chemicals, chemical formulas, um, I don't have those. Yeah, exactly. I'm staying from an optics perspective here. Staying focused on the optics class and not going too much into the bio. Just why we're only going to talk about parts of the eye that are relevant to optics and how we see, rather than all the other parts of the eye that like maintain each other. But yeah, okay. We're gonna start in 30 seconds. Let anyone else join in. Share my screen. There. This is what we're going to be starting with. And we have a preview of all the stuff we're going to be talking about today already. All right, so as I said, today we're going to be talking about the eye and color theory. So first thing we have that we're going to talk about is the iris. The iris is the colored part of your eye. When you have someone's eye color, that is what you're seeing is the iris. What's around the iris is called the sclera, and that's the white part. But the sclera doesn't play that much of a part in terms of optics. Rather, it's more of the iris. And one function of the iris is regulating the amount of light that is allowed into the inside of the eye, which, let me see if I can have a laser pointer here. Here, we have the drawing on here. The iris is this blue part, which does in part regulate by means of adjusting the lens, of adjusting the amount of light that gets in here, which we'll see what happens on the other side of the eye later. But yeah, that's the colored part of your eye, and the different pigments that the iris has determines the different color that you have for the eye. The iris is in between the cornea, which is right here, and the lens right here. So it's in between. It's on the outer side of the lens. Also, we do have a lot to get through today, so I'm going to be moving kind of fast. Let me know if I'm going too fast. So, second thing, the cornea. That's on the outside, and it's very, very important optically, because if you think about the eye like a lens, right, that takes parallel rays and bends them onto your retina, which we'll see later. That lens that we can think is comprised of two things: the cornea and the actual eye lens. That's what together makes up all of the ray bending power or optical power. The cornea is actually responsible for 2/3 of all that refractive power, and the lens is only responsible for 1/3 of the refractive power. So it's ironic that we call the lens because it actually does less lensing than the cornea itself. But yeah, fun fact, represents 1/6 of the outer layer of the eye. It refracts light. That's how it bends it, just like a normal lens. It refracts light when light shines on it, and then it's going to have a certain focal length. There's something called the near point of the eye for the lens and the cornea together. That's about 25 centimeters. That's basically the eye's focal length if you were to take it like a combined lens. Next thing, pupil. So the pupil is the opening in the middle of the iris that lets in the light. Adjusting the pupil size makes it so that you can have more or less light coming into the eye. The iris controls the widening and narrowing of the pupil, and so do the sphincter pupillae and dilator pupillae, which contract and dilate the pupil too. As you can see, dilator pupillae, we know that it dilates. It's pretty easy translation. But yeah, its function is purely just to regulate how much light goes in, and then it changes in size with light, this will see later, the process of light adaptation and emotion, actually. You'll have more dilated pupils in more intimate situations. That's a scientific fact. Next up is the lens. The lens is the other 1/3 of the refractive power compared to the cornea. And it's located directly behind the iris, just like the iris is located right in front of the lens. It's right here, labeled in the diagram. Right here, right? Iris and lens. And then it's a natural lens that we're all born with. There's actually a condition. I forget what it's named, but there's one atrocious condition where you can be born without eye lens. It looks pretty disgusting and unfortunate. I forget the name of it though. It'd be a cool thing to look up. But yeah, it focuses light rays onto the retina so that you can properly see the image so that you can properly make an image of what you're seeing and then translate that to the brain. The ciliary muscles attach onto the lens and they compress it or contract it so that we can change the focal length of the lens itself. And by changing the focal length, we can adjust where the image is going to be and if it's properly in focus with the retina. If you ever like bring something too close and you have to squint your eyes, what you're doing is you're changing the focal length of your lens, your total focal length of the eye, and you're basically just bringing it into focus so that you can see it properly. Does that make sense? Okay. So next, really quick point here. The ciliary body and muscles are the muscles that constrict and contract the lens, as we just said. And that allows the eye to focus on items that are either really close or really far. Sometimes you also have to squint your eyes a specific way to see something far or open them. That's just adjusting the focal length so that it can focus properly on your retina. So now let's talk about the back end of the eye. Here we have the retina. And this is where we're going to be focusing our image, this whole place right here. Technically not here for a reason that we'll see like in a second, but everywhere around here, we're going to be focusing the image, and then we're going to pick up that image and send it to the brain. But yeah, the retina is a light-sensitive layer that has photoreceptors, and these photoreceptors sense light, and then they create biochemical processes that make differences in potentials that get sent to the brain. And then the brain can read that image with visual processing, and that's a whole other topic. But yeah, it's composed of light-sensitive cells known as rod cells and cone cells, which are the two types of photoreceptor cells. It's basically just the film in a camera where you can put the image. That's what your eye has. Where you can put the film to get the real image. That's what's going on with your eye. You're trying to, with the lens, you're always trying to focus it so that the image appears directly on the retina. And if it does, then you have the clearest image possible with the highest visual acuity. But yeah, this is where you have to focus an image to see properly. If you're out of focus, then images will appear blurry or incomplete, and that's where we'll need corrections. So part of the retina is the macula, which is a more specified region at the back of the eye, right here. Right? And then that macula is more it's more dense in terms of the amount of cone cells that it has. For now, we haven't seen the differences between cone cells and rod cells, so hang on with me for a second. We'll get into those in a second. But for now, just remember that the macula has mostly cone cells and some rod cells. And the fovea, also known as the fovea centralis, has it basically has only cone cells. The population density for rod cells in your fovea is, I missed the one to the name. The population density for for rods in your fovea centralis is really, really low. But overall, the macula and the retina as a whole is populated with rods and cone cells, and rod cells are a little bit more scattered away from the fovea centralis, and it has an increasing population away from the fovea, while the cone cells are more concentrated around the fovea, and the highest population density for the cone cells are the fovea centralis. And then, as we said here earlier, there is the optic nerve with the optic disc. You can actually test this out yourself with an experiment on paper. I forget. If you write like the letter H twice on two sides, and then you cross your eyes and then look at it at a specific distance, you can actually see that the H has disappeared someplace because the image that you're seeing is focusing right onto the optic nerve. And we don't have photoreceptors in that region because that region is made for sending all of the information that we have from all of the photoreceptors around in the retina and sending them to the brain. So there's this very small disc in your eye that doesn't actually receive images. It doesn't sense images. And that's called the optic disc. It's also called the blind spot. And that's where your optic nerve is connected to your eye. And that optic nerve is responsible for carrying all the information to your brain. But yeah, we just said that there's a decreasing cone cell density from the fovea, and there's an increasing rod cell density from the fovea all the way to the retinal periphery, which is just like how far the retina extends. Everything makes sense so far? All right. Now let's look at some typical eye problems, the most basic ones, which are going to be myopia and hyperopia. So first of all, some terminology. An emmetropic eye is basically an ideal eye. It's an eye that doesn't need correction optically and focuses all of the images onto the correct focal plane, aka the retina, right? So usually, or for me at least, there are a lot of people who need glasses, and those are lenses. And why do we need lenses for glasses? Why do we need lenses for correction of our vision? Well, because sometimes the eye is a little bit longer or shorter than the ideal eye. And so when you have the lens in the cornea, their combined refractive power doesn't exactly focus the images at the correct place, aka it doesn't focus them on the retina. Instead, for myopia, it focused them in front of the retina, which means that when the rays get to the retina, it's blurry. It's not one focused image. And so when the photoreceptors pick it up and send it to the brain can't make much sense of that image. It's very blurry. And you won't see well. So the solution to having rays focusing in front of your eye is to put a converging lens. If you put a converging lens, these rays, which used to focus here, are now more diverging when they hit the eye. And so when the eye applies the same refractive power, they're going to bend further back. Does that concept make sense? If you bend the rays out a little bit more, it's going to take more distance for that same lens to focus them. So if you have more diverging lens, they'll focus at a farther point. And that's actually how you solve nearsightedness. If we think about it.
So far, you know.
Huh?
You are in the classmate, right?
Excuse me? Okay. I think that was accidental. So if we think about it, when an object the reason why this is called nearsightedness is that if one object is really close, say we have like a ball right here, right? These rays are going to be really divergent right here. They're not going to be parallel. So at that point, when we have diverging rays here, they're already diverging, so they'll focus on the right place. So when the objects are really close, you can still see correctly because the rays are divergent enough that the correction from your eye, the refractive power, makes them focus at the right place in the retina. But instead, when they're very far away, the rays will focus at the exact focal point of your combined lens system with your cornea and your lens, right? And that is going to be in front of the retina. And that's why we have the problem with nearsightedness and what we call it nearsightedness. We can see when an object is close, but not when it's far. And that is always fixed with a converging lens, a diverging lens. I'm losing my words today. The opposite case is also possible. Rays focus behind the actual retina, and the eyeball is too short or too small to properly focus an image. And for that means that this lens doesn't have the refractive power that it needs to focus it onto the retina. I forgot to ask earlier, but does the concept of refractive power make sense? If it has a higher power, it can bend more, so it has a smaller focal length. I think we went over that in class a little bit fast. Does anyone remember the units for refractive power? Diopters. No E at the end, just diopters. Spelled. Yeah, aphakia. That's the disease I was looking for earlier. Thank you. But yeah, so when it focuses behind the retina, we need something that's going to make the rays more convergent so that this lens is going to be enough for bending the rays to the correct spot. So if we put a converging lens in front here, it's going to bend the rays and make them more converging so that when we apply that same convergence, it's going to focus them at the right place on the retina. And so that is how you fix near farsightedness. And again, if we backtrack and think about why this is called farsightedness, is because if we have something that's infinitely far away, we're gonna be the closest to seeing it correctly because it's gonna be the closest here. But if we bring an object that is really close and has diverging rays, that image is gonna focus even farther back. So the farther it is, if you don't have enough refractive power, the farther it is, the better it's gonna be. And if it's closer, it's gonna be a lot worse because this grows a lot faster. Oopsies, that is not my eraser. So yeah, as a summary, myopia is fixed by diverging lens, and hyperopia is fixed by a converging lens. That's always the case. All right, so now let's talk about photoreceptors, and this is going to get kind of complicated fast, and you guys might not bear with me because it is a biochemical process that makes it so that we convert the stimulation of the photoreceptors all the way into an electrical or just a difference in potential that we're going to send to the brain to process the image that you're seeing. So photoreceptors, there are two types of them. We talked about this a little bit earlier. Some of them are rod cells and some of them are cone cells. We'll see what they do and why they're different later on. But basically, both types of cells, photoreceptors, have an outer segment which stays in the retina's pigmented layer. So that's the outer layer where it's going to be able to receive stimulation from the light rays. And then inside of these outer segments, there are some discs that contain visual pigments and photopigments. They get bleached by light, and then they get regenerated back into a cycle so they can get bleached again. And when they do get bleached, they make some biochemical process that basically gives the system as a whole a stimulation, which sends it on into the brain. So pigments get made, they get bleached, and they get regenerated, and they're in these like disc packets. The reason why they're on these disc packets is because it maximizes surface area that you can have the chemicals there. If you have more surface area, you're going to be more sensible. So they're in those disc packets just to maximize that surface area and be able to feel light better. But then also there's the inner segment, which is in the retina's neural layer. I didn't mention earlier that the retina had two layers, and that is the pigmented layer and the neural layer. Pigmented layer is very, very thin compared to the neural layer. It's actually like one cell thin. But yeah, I didn't think that was worth mentioning earlier because why complicate it when we can why complicate it when we can get to it now when it makes sense. And then under the inner segment, here I'm going to draw here a little bit. This is the outer segment for both, which it is drawn in the diagram, but I'm going to highlight it. And then this is the inner segment with the cell body down here, and then the synapses. So the inner segment is just in the retina's neural layer, and it does a lot of stuff. It just does a bunch of stuff with polarization of the cell for the biochemical process that we're going to talk about after. And then it's connected to a bipolar cell, which also has a biochemical process, which passes neurotransmitters to the ganglion cell, which then sends it to the brain. It's just electric signals because it's a difference in it's a very small difference in potential in millivolts. But yeah, that's the overall structure of the photoreceptors. And now let's talk about some chemistry. So the retinol is a molecule that can absorb light, and it pairs with opsin, which is a vitamin. It's very close to vitamin A. It combines with retinol to make visual pigment, and that visual pigment is what's going to get bleached. And the process of that getting bleached is it goes from a molecule called I think cis11 retinol to all trans retinol to breaking apart into retinol and opsin again, and then the retinol gets reused. It gets reused for the regeneration of the visual pigment for the discs. There are 4 types of visual pigments, and they have different wavelengths that they are bleached at. As I said, when the pigments get hit by light, biochemical process makes it so that it's bleached and that it changes shape. And then when it changes shape, that triggers some stuff. And then after a little bit of time, the retinol and opsin break apart, and then they're put back together again. It's a different opsin, but it's the same retinol molecule that can be used. And they're basically putting it in Q to make another disc. I'll mention that in the rod cells, the discs are like you could like take them apart if you used your hands, if they were small enough. But in the cone cells, they're collimated. There's actually you can't like take them apart one by one. They just have a disc structure. All right, here we go. Biochemical process. So basically, we have our 11 cis retinol, right? That when it's bleached, it basically stimulates this protein called G protein. I have no idea what that is, but it's a protein called transducin. And the transducin it activates an enzyme which is called PDE, phospho. I can't even read what that is. I just have it as PDE. And then that enzyme actually breaks down something called cyclic GMP, which is usually well, it maintains the channels of ions flowing through open so that the cell can stay not polarized. But when it's stimulated by light and 11 cis retinol becomes all trans retinol, meaning that it has been bleached, it actually breaks down the cyclic GMP. And so they're just GMP now, doesn't block, doesn't keep the channels closed, open. Instead, it closes them. It lets them close up, and thus after that, the inside of the cell is going to become polarized, in like electrically polarized with a plus side and a minus side, because the ions stop flowing in and out. That's the first part of the process. Even though what no I left at the bottom here long. But yeah, overall, in short, when the opsin changes shape, It makes an enzymatic cascade, which in the end results in the closing of the channels that lets the ions through when it's dark. When it's dark, it lets the ions through, and then as soon as you see light, it closes those valves, and then it doesn't let the ions through anymore. So this is just a step-by-step process of basically what happens right after that. So we just said that light activated pigments activates the G protein, and then the transducin, which is what we had last slide. Go back. Yep. The transducin here. There you go. That's the transducin. It activates the PDE, which starts breaking down the cyclic GMPs. I have no idea what a G protein is or what the PDE actually is in terms of the molecule or what cyclic GMP is. I just learned this from a textbook. And they're just molecules that you'll have to bear along with because they have no optical relevance except for carrying out these processes. And so I don't really know what they are apart from what they do. So when the cGMP is keeping the cells open, it depolarizes the cell and keeps it so the cell has a negative 40 millivolt dark potential. But when we close the channels by breaking down the cyclic GMP, it polarizes the cell. It doesn't let the ions through. And that is a difference in potential that's going to be negative 70 millivolts. So from that now, we have like a difference in potential that can measure if we feel light or not. If we're at negative 70 millivolts, we know that there's light that's detected by the photoreceptors. And if there's negative 40, we know that there is not light because the cell itself is not polarized. More polarized, more difference in potential. Does that make sense? Pigment doesn't get destroyed when you're receiving light. It gets bleached and then reused. But yeah, does this idea of polarizing the cell? Yeah, I'll post the slides and the recording after this. Does the concept of having a polarized, like an electrically polarized cell make sense? Like why when you're blocking the ions, it becomes there's more of a difference in potential. So that's kind of a harder concept. Okay, well, I'm not seeing any questions, so I'm going to assume that it makes sense. But yeah, when when the photoreceptors are unpolarized, there's going to be less potential, and when they're more polarized, there's going to be more potential. I know it's like I know negative 40 is more than negative 70, but you know what I mean. But yeah, as a whole, the whole process just changes the chemical. It makes the cell chemically polarized from receiving a stimulation from detecting light. More chemistry. So then what? So whenever the cell is not polarized, this potential in still the rod cell here is this is not polarized. So in the process of actually I'm just going to go to what happens when light hits. When light hits, this does polarize, which stops inhibitor neurotransmitters from leaving here into the ganglion cell. And here I'm realizing you can't actually see what I'm saying by here. But here, in the dark, since this is not polarized, this is going to have some IPSPs, which are just they're small processes that are used specifically by these cells to it's not like an action process which gets sent to the brain, but it's like a smaller difference in potential that means something more specifically to that cell. And so when the cone cell itself or the rod cell is not polarized, that causes the bipolar cell to be hyperpolarized. And when it is hyperpolarized, it releases inhibitor neurotransmitters, which essentially don't let the ganglion cell produce a signal. But when we have light, now this becomes polarized. So this unpolarizes. It's not polarized anymore. And so the lack of IPSPs is going to cause the lack of polarization. Please excuse me. And then that is going to allow the release of neurotransmitters into the ganglion cell, which is going to produce a signal out here that we can see. And that is sent to the brain as essentially an electrical signal because it's the difference in potential. Technically chemical potential, but you get what I mean. This is a very big overview of what happens. It's very, very simplified because I could talk about this for an hour probably. All right. So now we're going to come back to the overall big picture of photoreceptors, and we're going to look at them as a whole. So there are two types of photoreceptors. Some of them are called rod cells, and the others are cone cells. Why? Because they look like rods and cones. Simple naming. But the rod cells, they're the ones that are responsible for black and white vision. They're more sensitive than cone cells. So they're the ones that you use in the dark, or they're the dominant ones in the dark, the ones that represent more of your visual acuity in the dark, because they're more easily stimulated, and so they work in dim light. They're actually closer to the outside of the retina than the cone cells because they're like taller, so they're deeper inside of the retina from the outside, which means that they're closer from the inside, and so that partly explains why they're more sensitive to light. But yeah, they work better in dim light, and they're your main producers of vision when there isn't light or when you're in the dark. And with respect to that, they're also, since we said that they're more popular, or their density is greater when you get away from the fovea, which is essentially like the center of your vision, center of your eye. They're more commonly found in peripheral of the retina, so they're going to be responsible for more of the peripheral vision. And we know that our peripherals don't produce as good of or as clear as an image as the center of what we see. That is because the rod cells don't produce that good an image. They're meant to be really sensitive and work in the dark rather than producing good visual acuity. So they're going to give you blurry and non-sharp images. But yeah, that's why when you have dim light, if you go to really dim light, you'll have color disappear from the edges of your vision, and these objects are going to look fuzzy because it's going to be picked up by only your rod cells. And as we said, the rod cells have really bad visual acuity, and they work in black and white. The visual pigment for rod cells is called rhodopsin, and it is bleached the best at 700 nanometers. That's an important fact that we'll look at later. Next are cone cells. Cone cells are the ones that are responsible for your color vision, and they're a lot less sensitive than, oh, this is supposed to be rod cells. That's my bad. They're a lot less sensitive than rod cells. And so they're less stimulated, and they only work well in bright light when you're just during the day. the cone cells are what's responsible for most of your visual acuity by very far. They are the ones that produce a clear colored image. And they are found mostly in the fovea and the macula, as we've said before. Instead of rhodopsin, they have a pigment called iodopsin, which is, there's actually 3 types of that, one for each primary color of an additive color system, which we'll see later. But yeah, they're a bit deeper in the retina than the cone cells, so they're not as sensitive because they're farther away. And as I said, there are 3 types of cone cells, because of their pigments. And there are L cone cells, which are just long cone cells, and they are for red, which makes sense because red has the longest wavelength. And those are the most common types of cone cell. Energy-wise, it also makes sense. Then we have medium cone cells, which are the cone cells for green. A peak wavelength of about 530 nanometers. These aren't fully accurate. They're like around 530 because I believe it changes a little bit between every person. It's not really standard. There's not really a way to measure that too well. Those are just common. They're not the most common or the least common. And again, energy-wise, it makes sense. And there's S cone cells. And then S cone cells are the least common by far. And they're responsible for blue color. And the peak wavelength, the wavelength that it bleaches pigment at the most, is 420 nanometers, around there. Here's a quick diagram of how we can visualize the wavelengths of the cone cells. This is basically relative stimulation of the cone cells when you have different colors of light. And so down here we have our different colors of light and the responses to all 3 types of cone cells. So we see, we said the blue cone cell peaks at 420. That's about right. Then we had, what was it? 530 and 560. First peak, second peak. That's about it. You'll find different values online, I'm sure, if you look it up. But that's about what they get most stimulated by. So here's a quick table for comparison of rod cells and cone cells. Rods, no color. Cones, yes color. And in fact, 3 colors. Rods, high sensitivity. Cones, very low sensitivity. So they operate in bright light better compared to rod cells who operate in dim light. Rods have low acuity. Cones have very high acuity. And then rod cells are actually a lot more numerous than cone cells. There's around 20 rod cells for each cone cell that there is in your eye. And then the population density of rod cells and cone cells. Rods are mostly in the peripheral retina because their density decreases as you get closer to the fovea. All right, light adaptation. So you guys have definitely just walked from a very dark room to a very bright room or turned on the lights at night, and then your eyes just basically don't respond for a couple seconds. And that's because you're adapting to light. So what happens is that when you're in light, when there's no light and you're in a dark room, your eyes are adapted to the dark. And that's not exactly the same thing as when they're adapted to when there's a lot of light, because in the dark, the rod cell system is a lot more active because there's not much light and they work better in dim light compared to the cone cells, which work better in bright light. And so when there's not bright light, they basically don't do anything. So when you're going for from dark to light, your rod systems suddenly just gets overstimulated. It makes sense that since they're very sensitive, when you shine a lot of light at them with a lot of intensity, it just basically goes crazy. There's light everywhere. And so with that, if we just kept those simulations, you basically couldn't see anything. Rather, the body, or specifically the eye, turns off that system for a little bit until you get back to the dark. The rhodopsin, that's inside of the sorry, that's supposed to say transducin. Again, my mistake. The transducin, which is that one enzyme that we had. Transducin. The transducin that we had that activates the enzyme that breaks down the cGMP, it moves to the inner segment of the cone cell, or sorry, of the rod cell, and so that inhibits the process of transduction, which is process of seeing light and converting that to a signal. It just stops that process completely by moving the transducin to the inner segment. And the cones are also desensitized a little bit, but they become dominant for vision. They do not desensitize too much. And they now play a greater role in determining what you're seeing. So overall, the light adaptation process is a loss of retinal sensitivity, but you're gaining visual acuity. So it's a trade-off. Also, when you're adapting to light, your pupil size is going to change because you're going to allow less light into your eye to dampen the intensity. And so your pupillary reflexes, which are just muscles around your pupil, they contract the pupil to allow less light, and thus you're going to have less intensity. When you're adapted to the dark, it's the exact opposite from that. And the one small difference is that it takes a lot longer for the transducin to move back from the inner segment to the outer segment. So that's why it takes like around 20 minutes compared to like a couple seconds. It's like a fast deactivation versus slow reactivation, which if you think about it also makes sense. All right, let's talk about the final part of this lesson. I know I've been talking for 45 minutes now and you guys are probably a little bored, but we're almost there. Finally, let's talk about color. And color has three fundamental properties. Those are hue, value, and saturation. Hue is like the color family that a color pertains to, whether that be just red, blue, green, or any color in between. It's the overall color family. And there's value. Value is basically where it is on a grayscale point of view. It's how dark or how light is it. If you want value, you have two graphs of it, like here and here. This is what we mean by value, how light or dark it is. And finally, there's saturation. Or also called intensity, which is just how bright that color is or how color concentrated. Here, it's very not bright. There's not that much of the color there. It's like making solutions in chemistry. We have very concentrated versus not concentrated solutions, but that's a bad analogy, I guess. Here, let me use another color because we can't see red on red. This is a very high saturation. High. And this is a very low saturation. There's not that much of that color there. Yeah, those are the three fundamental properties of color. You'll definitely see that pop up in a bunch of places for Science Olympiad. Finally, color theory. There are a bunch of color models, but the two most used ones are additive color theory and subtractive color theory. Now, additive color theory comes from three primary colors, red, green, and blue. And from those three colors, we can make every single other color. Starting with cyan, yellow, and magenta. Those are going to be our secondary colors for this color model. You'll see on the next slide why I included the K, because it's actually part of the middle, all of the combinations. But here it would be white in the middle. I'll note that when you have light, when you have every single color, that makes white light. And when you have no light at all, that's black light, or just no light, right? In subtractive color theory, it's a little bit different. When you have every single color, you make black, but when you have no color, it's just white. But for additive color theory, it's the opposite of that. All light is always white. As I said, the additive color theory model is always used for light. It's used for predicting what color you're going to get when you combine two rays of light. When you combine red and blue, light, not pigments, light. Light is additive color theory. That's very important. And an application of that is photography. If you've ever seen, if you've ever photoshopped a little bit, and then you have this one picture, and this one guy, he looks a little green, so you add in a little magenta to make that a little bit brighter. That's one way that you can adjust an image, but then you shouldn't do it too much, because then everyone else in that picture can look pink, right? So these colors, which are on opposite sides of the color wheel, so magenta and green, cyan and red, and then yellow and blue, they're basically color opposites. When you have one, that's all minus the other. So yellow, if you were to write an equation form, yellow is like all light, which is white light, minus the blue light, which is the blue component. Why? Because when we have all three, we're in white light. We are right here on the color wheel. But when we take out the blue light, then we go this way. That's how the opposites work. And if you think about it logically, when we have blue, red, and green, and we take out blue, we're left with just red and green. And that's just yellow. But yeah, you can sort of make these small equations with the additive color theory when you're going to predict what type of color you're going to get from combining multiple rays of light. The other fundamental color theory model is the subtractive color theory. And this one is used very widely for printing. You'll see that the colors of ink from your printer are not they're not red, green, and blue. They're cyan, magenta, and yellow. And then you'll have black, which is a combination of the three. So here our primary and secondary colors are inversed. Like you can see that in our previous diagram, we had red, green, and blue, and then cyan, magenta, and yellow. And now it's the opposite. Our primary colors are cyan, magenta, and yellow, and we have red, green, and blue on the inside. And again, it still makes sense because they're a combination of each other. So when you invert it, it's still going to have that fundamental relationship. But now the outside, when you don't have these colors, is going to be white. And the inside, when you have all these colors, is going to be black. Because the point of this color theory model is saying that when you're adding cyan, you're taking away red. Because it's the opposite of red. So when you add cyan, you take out red. When you add green, when you add magenta, you take out green. And when you add yellow, that takes out some of the blue. So when you have cyan, magenta, and yellow, you've taken out all of the colors, aka all the primary colors. And so you're left with no light. but this isn't exactly for light. This is meant for pigments. And so it's frequently used for dyes, coloring, and then ink, which is what printers have. And that sort of makes sense when you think about it. The mindset of subtractive color theory makes sense when you see something. Why? Because when something looks green, the reason why the grass is green is because it absorbs every other color but green. The only thing that reflects from you is green. So in that sense, subtractive color theory does make sense for real life, and so it makes sense for pigments, which they're not the same as one important distinction is that they're not the same as the pigments we talked about earlier. The photopigments, that's not the type of pigment I mean, but I'm sure you know what I mean when I pointed out dyes and inks. But yeah, does that make sense? Why we would have subtractive color theory for things in real life that are not directly light? Can I get a sign of life after I talk for an hour? Okay. Well, final slide, and it looks like we're on time. I know I talked a lot today, but this is an extra lesson for extra content. So, I mean, I wasn't going to do some crazy interactive activity. But yeah, there are many color models. We've just talked about two, which are the two most fundamental ones. But they all have their own use. Additive is used for light combination. Subtractive is used for printing. HSV, it's used for histograms and for converting to grayscale. Why? Because it's represents color in terms of their three properties: hue, saturation, and value. That's what HSV stands for. It's a very clever way of representing colors. And then YIQ, which is used for TV broadcasting, and there's like CIE, which is used for math, and then human perception. There's a bunch of these if you go to Wikipedia. A bunch of models that you can read about their uses, but they're not really relevant. Do you guys have any questions? Something I went too fast on? We are officially done the contents of this class. Any questions at all? Any comments? Any fun facts? How many color models? More than you can count on your fingers. There's not one definite amount of color models because people make color models to represent colors different ways. So you could make your own color model if you were if you wanted to code an application that represented color differently, you could, and then you'd have your own color model if you wanted to. So you can make as many as you want. It's just a way to represent color. There's not a definite amount of color models, and they keep making more for new uses. Does that make sense? All right. Well, as I said, I'm done talking for the day. If you guys have any questions, feel free to ask, and then if you don't, you're off the hook.
Bye. Thank you so much.
No problem. My pleasure.